Hello, my name is Kartik. I am a high school student in Cupertino, California, and today I'll be presenting my research project on gene expression analysis of myeloproliferative neoplasms that I conducted with the guidance from my research mentor, Dr. Melanie. I'll begin with some background information on myeloproliferative neoplasms, then I'll share my research objectives, walk through the methods, highlight key results, and finally end with conclusions, significance, and future directions. Let's start with the background. So what are myeloproliferative neoplasms? Well, MPNs, which is the abbreviation, are a group of rare blood cancers that are characterized by an abnormal production of blood cells in the bone marrow, including red blood cells, white blood cells, and platelets. In these images, we can see the difference between healthy hematopoietic stem cell differentiation and cancerous or abnormal differentiation of these cells into myeloid and lymphoid cells. In MPNs, we see abnormal differentiation and proliferation of these stem cells, causing excessive over or underproduction of these cell types, as well as producing dysfunctional cells. There are four main subtypes of MPNs I focused on in this study. Polycythemia vera, abbreviated as PV, causes an overproduction of erythrocytes or red blood cells. Essential thrombocythemia, or ET, is the overproduction of thrombocytes, which are platelets. Primary myelofibrosis, or PMF, involves scarring in the bone marrow itself, resulting in too few blood cells being produced. PMF occurs without pre-existing early-stage MPNs, so it develops independently. On the other hand, secondary myelofibrosis, or SMF, happens when an early-stage MPN like PV or ET transitions into myelofibrosis. These diseases are strongly associated with mutations in the JAK-STAT pathway. The JAK-STAT pathway is a series of transmitted cytokines and growth factors that control blood cell growth and division. The most common MPN mutation, the JAK2 V617F mutation, causes continuous activation of signaling in this pathway that normally regulates blood cell growth. This results in uncontrolled proliferation. Across subtypes, patients can experience panmyelosis, or the overproduction of all major blood cell types, splenomegaly, which is the enlarged spleen, and thrombosis, an increased risk of clotting or bleeding. General symptoms like fatigue, fever, and weight loss are also common. Now I will discuss some objectives of my research study. Previous research, such as a study by Baumeister et al., has looked at gene expression in MPNs, but only by focusing on just one subtype at a time. This leaves a big gap. We don't fully understand how gene expression patterns differ between the stages or how MPNs progress. My project set out to address that gap. Specifically, I wanted to identify stage-specific biomarkers of MPNs, compare gene expression across the subtypes, and enable earlier prediction of progression from early-stage MPNs into myelofibrosis. To meet those objectives, here's the methodology that I used. I followed 5 steps. First, collecting CD34 plus cell microarray data, then processing and normalization, then identifying differentially expressed genes, gene annotation and visualization, and finally performing gene ontology enrichment analysis. First, I found the data I used for my own analysis, which was deposited in NCBI's geodatabase, accession number GSC 174060. The original experiment, which produced the raw microarray data, was conducted by Baumeister et al. At the Department of Hematology at the University of Aachen. The patient data came from the University of Aachen and included 36 samples, 6 ET, 11 PV, 4 SMF, 9 PMF, and 6 healthy controls. The methods that this research group used to collect the transcriptomic profiles from each patient sample in their original experiment is listed here. First, a FICAL pack premium kit was used to isolate the mononuclear peripheral blood and bone marrow cells. Then, magnetic cell separation and enrichment of the CD34 plus hematopoietic stem and progenitor cells was done using a microbead, kit. Next, 32 genes associated with hematological cancers were targeted with next-generation sequencing. And finally, the RNA transcripts were extracted and transcriptomic profiling was conducted with Affymetrix HTA 2.0 microarray platform, and then the results were deposited in NCBI. After downloading the raw cell files of the microarray data, I preprocessed the data in R using the oligo package. With the robust multi-array method, I applied background correction, normalization, and expression calculation. Normalization is essentially adjusting the data so that all the samples are comparable, removing technical variation that isn't biological. These box plots confirmed that the samples were successfully normalized and comparable. As you can see in Figure 1, before normalization, the plots were uneven. But after normalization, as you can see in Figure 2, they line up at the same median, which confirms that the samples are standardized and can be reliably compared during analysis. Next, I used the lemma package in R for differential expression analysis. This essentially is comparing two conditions levels of gene expression to find genes that are significantly over or underexpressed. These identified genes are differentially expressed and are called DEGs. I created a design matrix, fit linear models, and defined contrasts to compare each MPN subtype against healthy controls. DEGs with adjusted p-values of below 0.05 were considered to be significant. After identifying the DEGs, each list of DEGs was annotated with gene symbols and Entrée IDs using the HTA20 transcript cluster database. The results were then visualized with several kinds of plots and charts. For example, in Figure 3 here, you can see the first 12 rows of an annotated gene list, which includes log-fold change and p-values, which indicate statistical significance or the chance of the data point being generated due to random chance or technical error. Finally, I used the gProfiler 2 package to perform gene ontology enrichment analysis. This helped identify which biological pathways and processes were affected by the DEGs in each subtype. Again, the significance threshold was a p-value less than 0.05, and the resulting data was visualized with a ghost plot. Now let's look at the results. First, differential expression analysis showed large differences between the subtypes as shown through this bar chart in Figure 4. The number of DEGs from most to least was PMF, then SMF, PV, and then finally ET. This shows that PMF had the greatest degree of dysregulation, while ET had the least. Here we have a heat map in Figure 5, which displays hierarchical clustering of the most differentially expressed genes between all subtypes of MPNs and healthy controls. Red indicates high gene expression and blue low gene expression. The heat map reveals distinct clustering. Secondary myelofibrosis had a very different gene expression profile compared to the other groups, while PV and ET clustered closely together as expected. This suggests that ET and PV are molecularly similar, while SMF diverges significantly. Starting with primary myelofibrosis, I will discuss the results and significance of the Go enrichment analysis of each comparison. Here's Figure 6. We can see that PMF had both the greatest number of DEGs and the highest statistical significance of the results. The most enriched categories, which had the tallest bubbles or the highest negative log of the adjusted p-value, included structural constituents of chromatin, nucleosome assembly, and DNA methylation. These all point to chromatin remodeling and epigenetic modifications as major features of PMF progression. Next, we will look at the largest bubbles on the plot, which represent categories with the greatest number of enriched genes. PMF showed a high enrichment of genes linked to cytogenetic abnormalities, protein dimerization, and protein heterodimerization, processes that are central to JAK-STAT signaling. Interestingly, keratinocyte-related genes were also enriched, which suggests a possible link to fibrosis, mechanisms observed in other tissues like skin fibrosis. Next, secondary myelofibrosis showed the second highest number of DEGs. But as you can see in figure 7, SMF versus a healthy control comparison, DEGs had lower significance than PMF. The most enriched pathways included toll-like receptor signaling and interleukin-10 signaling. Both of these address inflammation in the body. On one hand, toll-like receptor signaling, which shows the strongest significance, is responsible for detecting pathogens and precipitating pro-inflammatory pathways. Their abnormal activation in the bone marrow has been proven to contribute to fibrosis. On the other hand, interleukin or IL-10 signaling is responsible for mitigating these pro-inflammatory pathways and immune responses, and it is a core signaling process in the JAK-STAT pathway that regulates fibrosis. Its activation can be seen as an effort by the immune system to compensate for the overactivation of the JAK-STAT pathway in many MPNs, and as well as here in SMF. Next, the categories for SMF versus healthy controls with the greatest number of DEGs included activator activity and cytokine receptor binding, which was the largest category and the most significantly enriched category. The enrichment of genes associated with activator activity indicates that gene expression regulation is a central part of SMF, which complements the cytokine receptors and other signaling molecules that have been found to be impacted in SMF. Finally, Go molecular function cytokine receptor binding was one of the most statistically significant categories and had a significantly high number of genes that encode cytokine receptors, which were found to be dysregulated in SMF. Cytokine receptors are crucial to the JAK-STAT pathway, activation, and inflammatory signaling. Its overexpression can lead to the bone marrow fibrosis and chronic inflammation, which is commonly seen in MPNs. Next, we will look at polycythemia vera, which displayed fewer enriched pathways than PMF or SMF. The strongest enrichments came from microRNAs, which specific transcripts are listed here. These microRNAs regulate tumor suppressor genes like phosphatase and tensin homolog, and cytokine suppressors like SOX, both of which normally limit JAK-STAT activation. Their dysregulation suggests microRNA involvement is key to PV progression. Essential thrombocythemia had the fewest DEGs, with only one gene significantly upregulated compared to controls. Additionally, no Go pathways reached statistical significance. This shows that ET has the mildest molecular dysregulation among the subtypes. Now for the conclusion. Each MPN subtype has distinct molecular signatures. PMF is dominated by chromatin remodeling and cytogenetic abnormalities. SMF is defined by cytokine signaling and inflammation. PV shows dysregulated microRNAs, and ET shows very minimal molecular changes. However, despite their differences, one theme was consistent among all the MPN subtypes, and that was JAK-STAT pathway dysregulation, which confirmed its central role in MPN pathogenesis. These results suggest predictive biomarkers for progression from early-stage MPNs into fibrosis. For example, cytokine receptors, toll-like receptors, and interleukin signaling components could be monitored in PV or ET patients via frequent blood-based assays to detect these surface proteins and signaling molecules, which could help identify early signs of progression to SMF. Looking ahead, this research could help develop personal risk scores to predict progression of early-stage myeloproliferative neoplasms like polycythemia vera and essential thrombocythemia's progression to later-stage MPNs such as secondary myelofibrosis. This would help precipitate earlier intervention and design personalized therapies based on transcriptomic biomarkers, which would ultimately improve qualities of life of MPN patients and outcomes. These are the references that supported my analysis. If you would like to learn more about the background information on MPNs, my sources are listed here. And with that, I'd like to thank you all for your time and attention today.